Here is something strange to consider before you do anything else tonight. The molecule that is, at this very moment, coiled inside almost every one of your cells is older than language, older than bone, older than the idea of you. It was already ancient when the first fish crawled onto land. It does not know it is inside you. It does not know anything at all. And yet, somehow, it remembers everything that ever happened to your ancestors going back roughly 3.5 billion years. Not as stories, not as memories, but as a as a kind of silence that simply keeps repeating itself, generation after generation, in the dark. You don't have to do anything right now. You are already holding the oldest archive in existence, and it is humming along quietly, copying itself in your skin, your gut, the roots of your hair, without asking your permission and without needing your attention. Right now, in this exact moment, somewhere in your body, an old cell is finishing its work, and a new one is taking its place. And inside that new cell, the same long, twisted instructions are unfurling themselves and being read all over again, patiently, automatically, the way a tide doesn't need to be told to come in. You don't have to understand any of this for it to keep happening. You don't have to be awake for it. It was happening while you were a child, half asleep on a car ride home. And it will be happening tonight, in the hours after this story ends and your thoughts finally let go of their shape. So let your attention go soft instead. Let your eyes close if they haven't already. Tonight, we're going to travel into the strange, slow story of how human beings figured out that this archive even existed, how they stumbled toward it in the dark, the way you are lying in the dark right now, and how, piece by piece, decade by decade, they began to read it. It begins, strangely enough, not in a gleaming laboratory, but in a pile of used surgical bandages. In the year 1869, in the German university town of Tübingen, a young Swiss physician named Friedrich Miescher was trying to understand the chemistry of the cell, and in particular, the chemistry of white blood cells. White blood cells were difficult to get in any quantity, except in one rather unglamorous place, the pus that collected on bandages from a nearby surgical clinic. So Miescher collected those soiled bandages, washed the pus from them, and began breaking the cells apart, trying to isolate whatever was hiding inside their nuclei. What he found was a substance unlike anything chemists had cataloged before, rich in phosphorus, resistant to the enzymes that broke down proteins, faintly acidic. He called it nuclein because it came from the nucleus. He had, without fully realizing it, become the first person in recorded history to isolate DNA. Think about that for a moment. The molecule that would eventually rewrite biology, medicine, criminal justice, our understanding of disease and ancestry, and the tree of life itself. It was first pulled out of bandage pus by a young man working largely alone in a dim room, almost by accident. Miescher hadn't even set out to study heredity. He'd originally trained as a physician, following something of a family tradition. His father and uncle were both respected doctors, but he found himself drawn instead toward the quieter, more exacting questions of cell chemistry, working in the laboratory of a researcher named Felix Hop Saylor. The work was unglamorous in almost every sense. Day after day, he processed buckets of pus, collected from a local surgical clinic, washing and filtering, and treating it with various chemicals, watching for whatever the cells inside might be hiding. When he finally isolated his strange new substance, he noticed it behaved unlike anything chemists had described. It didn't dissolve the way proteins did. It resisted the digestive enzymes that broke other molecules apart, and it released far more phosphorus than any known biological compound. He called it nuclein because it seemed to live specifically inside the cell's nucleus, the small, dense compartment most cell biologists at the time considered structurally important but functionally mysterious. Miescher himself wasn't sure what he'd found. He suspected it might be important to heredity. In his private letters, later collected and published by his family, he speculated, cautiously, that this strange acidic substance might somehow be connected to how traits passed from parent to child, perhaps through some kind of complex chemical arrangement, almost like a coded alphabet. It is a striking thing to read those letters now, knowing what we know. Miescher's reaching, almost blindly, toward an idea that wouldn't be confirmed for another 75 years. But he had no way to test that idea, and the tools of his era couldn't take him any further. He spent the rest of his career returning to nuclein, fascinated and frustrated in roughly equal measure, never quite breaking through to what it meant. Occasionally setting the work aside for years at a time to study other things, the way a person might return again and again to a locked door without ever quite finding the right key. He died in 1900 of tuberculosis, still uncertain, never knowing that the substance bearing his original name would eventually be renamed deoxyribonucleic acid and would come to be recognized as perhaps the single most consequential molecule in the history of biology. And here's where it gets interesting. At almost exactly the same time, Miescher was washing bandages in Tübingen. A quiet Augustinian friar in a monastery garden in Brno, in what is now the Czech Republic, was running an entirely different kind of experiment, one that had nothing, on the surface, to do with nuclein at all. His name was Gregor Mendel. He had entered the monastery, by his own later account, partly because it offered him a path to education and a stable life he couldn't otherwise have afforded, having grown up the son of a peasant farmer in a small Moravian village. He had tried, and largely struggled, to pass the examinations required to become a certified teacher, and it was in the monastery's quiet gardens, and with the encouragement of the abbot, who had a strong interest in the natural sciences, that he found the freedom to pursue a long, methodical experiment most people around him would have found almost absurdly tedious. Between 1856 and 1863, he grew and crossbred thousands of pea plants, tracking traits like seed shape and flower color across generations with the patience of a man who had nowhere else he needed to be. Peas were, in a sense, the perfect subject. Easy to grow in large numbers, simple to control for cross-pollination, and blessed with traits, like round versus wrinkled seeds, that came in clear, distinct categories rather than a vague blur of in-between possibilities. He counted. He recorded. He found patterns, ratios that repeated themselves with uncanny regularity, generation after generation, as though something discrete and countable, rather than some vague blended essence, was being passed from parent to offspring, where earlier naturalists had often expected inherited traits to blend together smoothly, like mixing two paints into a new shade. Mendel found something closer to a set of switches passed down in predictable mathematical proportions, roughly 3 to 1, in certain crosses, a ratio that kept reappearing no matter which trait he tracked. He published his findings in 1866 in the relatively obscure proceedings of a local natural history society. And then almost nothing happened. The scientific world of his day mostly ignored him. His paper sat quietly in academic journals, cited by almost no one, for some 34 years, while Mendel himself, increasingly absorbed by his administrative duties, after being elected abbot of his monastery in 1868, largely set the work aside. He died in 1884 without ever knowing that the patterns he had so carefully counted in his garden would one day be considered one of the founding observations of modern genetics. It's a strange thing to sit with, that the basic mathematical rules of heredity were sitting in print, fully discovered, decades before anyone understood the physical molecule that carried them out. Two halves of the same enormous puzzle, born in the same years, in nearby parts of Europe, with absolutely no idea the other one existed. Mendel had found the rules of the game. Miescher had found, without knowing it, one of the pieces on the board. It would take other people, other decades, to realize the two discoveries were even talking about the same thing. Mendel's work was finally rediscovered around 1900, when three botanists working independently, Hugo de Vries in the Netherlands, Karl Correns in Germany, and Erich von Tschermak in Austria, each arrived at similar conclusions about inheritance, and then, digging through the older literature, found that a forgotten monk had already done the work decades earlier. Suddenly, Mendelian inheritance had a name and a growing audience. But there was still a missing link. Scientists now had rules for how traits were inherited, and they had Miescher's strange nuclein sitting unexplained in the nucleus, but almost nobody thought the two were connected. Most biologists in the early 1900s assumed that proteins, not nucleic acids, were the molecules of heredity. Proteins were structurally complicated, built from 20 different building blocks in endless combinations. Nuclein, what we'd now call DNA, seemed by comparison almost insultingly simple. This belief had a name and a face. A biochemist named Phoebus Levene, working at the Rockefeller Institute in New York through the early decades of the 20th century, carefully identified the basic chemical components of DNA: a sugar called deoxyribose, a phosphate group, and four nitrogen-containing bases named adenine, thymine, guanine, and cytosine. This was, in its own right, careful and valuable chemistry. Levene was a meticulous experimentalist, and his work mapping out the basic building blocks of nucleic acids gave later scientists the vocabulary they needed. But Levene also proposed something called the tetranucleotide hypothesis. The idea that these four bases simply repeated in a fixed, monotonous sequence over and over, like a four-note tune played on a loop. A molecule like that, he reasoned, could not possibly hold enough information to build something as complicated as a living organism. It would be like trying to write all of human literature using only four letters, each one appearing in the same unchanging order. Surely, scientists believed the real instructions for life had to live somewhere more sophisticated. It's worth pausing on how reasonable that conclusion seemed at the time, even though it would later turn out to be wrong. Proteins really were structurally elaborate. 20 different amino acid building blocks arranged in long, varied chains that folded into complicated three-dimensional shapes, each shape suited to a particular biological job. DNA, by comparison, seemed almost embarrassingly plain. 4 letters, an apparently repetitive backbone, nothing that looked, on the surface, like a sophisticated information storage system. Science doesn't always advance by brilliant new evidence overturning old ideas in a single decisive moment. Sometimes it advances by an old, comfortable assumption simply sitting unexamined for decades, blocking the obvious next question, until a slow accumulation of smaller findings finally makes it impossible to ignore. So for decades, DNA was treated as a kind of cellular scaffolding, structurally present, biologically uninteresting. The protein was assumed to be where the real secrets were kept. It would take a quietly persistent set of experiments conducted across 2 continents and nearly 2 decades to undo that assumption. The first crack came from a British medical officer named Frederick Griffith, working in London in 1928, studying pneumonia at a time when the disease still killed a significant share of the people it infected, well before antibiotics existed to treat it. Griffith was investigating Streptococcus pneumoniae, which comes in 2 strains, a smooth, disease-causing strain whose colonies appeared glossy under the microscope because of a protective outer coating, and a rough, harmless one, lacking that coating and lacking, it seemed, the ability to cause illness on its own. In a now famous experiment, Griffith injected mice with heat-killed smooth bacteria, which on their own caused no illness, mixed together with live harmless rough bacteria. Logically, by every assumption of the time, neither should have hurt the mice. The smooth bacteria were dead, and the rough bacteria were harmless. And yet the mice died. When Griffith examined their blood afterward, he found living smooth, disease-causing bacteria, despite having only ever injected a harmless living strain alongside a dead dangerous one. Somehow, something from the dead, disease-causing bacteria had transferred into the living harmless ones and made them dangerous. And that change was passed on to their descendants too, holding stable across generations of bacteria long after the original dead cells were gone. Griffith called this mysterious process transformation. He had no idea what the transforming substance actually was. He'd found a ghost, but not yet a name for it. And by most accounts, he remained a cautious, almost reluctant interpreter of his own remarkable result, never pushing the implications further than the data could strictly support. That question sat unanswered for 16 years, until a trio of researchers at the Rockefeller Institute, Oswald, Avery, Colin MacLeod, and Macklin McCarty, set out to identify exactly what Griffith's transforming substance was made of. Working carefully through the early 1940s, they took the heat-killed disease-causing bacteria and began systematically destroying different classes of molecules within it. First, proteins, then fats, then carbohydrates, then RNA, to see which removal would stop the transformation from happening. Each time, transformation still occurred. Then, they targeted the DNA itself, using an enzyme that specifically broke it down. And the transformation stopped completely. In 1944, they published their results. It was DNA, not protein, that carried Griffith's mysterious hereditary message. Avery was characteristically cautious in how he stated the finding. He was, by most accounts, a careful and somewhat shy man, deeply aware of how radical the implication was, and he avoided overclaiming in the paper itself. But the data spoke plainly enough. The molecule that biologists had dismissed as too simple to matter was, in fact, the very thing that carried the blueprint of life. Not everyone believed it right away. Old ideas don't fall the moment new evidence arrives. They erode slowly, argument by argument. Some scientists suspected lingering protein contamination might be responsible for Avery's results. It took a second, more dramatic experiment to truly close the case. In 1952, on Long Island, New York, two researchers named Alfred Hershey and Martha Chase devised an elegant test using viruses called bacteriophages, viruses that infect bacteria by injecting their genetic material into the bacterial cell while their outer protein shell stays behind. Like a syringe left on the doorstep, the phage itself is a strange, almost mechanical-looking thing under a microscope, with a geometric head and a thin tail that latches onto the surface of a bacterium like a key finding a lock. Hershey and Chase used radioactive labeling to tag the virus's protein with one isotope, sulfur, and its DNA with another, phosphorus. Since protein contains sulfur but essentially no phosphorus, while DNA contains phosphorus but no sulfur, a small chemical quirk that let them track the two molecules separately. They let the labeled viruses infect bacteria, waited just long enough for infection to take hold, and then used an ordinary kitchen blender to violently shake the empty viral shells loose from the outside of the bacterial cells. A step so simple, it almost feels like a joke buried inside a serious experiment. When they checked which radioactive material had actually entered the bacteria and gone on to direct the production of new viruses inside them, it was the phosphorus-tagged DNA, not the sulfur-tagged protein. The protein shells, knocked off by the blender, never made it inside at all. The Hershey-Chase experiment, as it came to be known, settled the argument for most of the remaining skeptics. DNA was the molecule of heredity. Now scientists needed to know what it actually looked like. This is where the story slows down and becomes, in a way, more human, full of rivalry, brilliance, frustration, and a few choices history has spent decades quietly re-examining. By the early 1950s, several research groups were racing toward the same prize: the physical structure of DNA. At King's College London, a physicist turned biologist named Maurice Wilkins was using a technique called X-ray crystallography to study DNA fibers, firing X-rays at purified strands of the molecule and analyzing the diffraction patterns left behind. A bit like reading the shadow a complicated object casts rather than looking at the object directly. In 1951, a brilliant and exacting young chemist named Rosalind Franklin joined the same lab to lead this work. Franklin had already built a formidable reputation studying the structure of coal using X-ray techniques in Paris, and she brought a rigor to the DNA crystallography that hadn't existed before, refining the equipment, controlling humidity precisely, producing the sharpest diffraction images of DNA anyone had yet achieved. The relationship between Franklin and Wilkins, by most historical accounts, was strained from very early on. Partly a matter of crossed expectations about who was meant to be leading the project, partly simple personality friction. They worked in something closer to parallel than partnership. Meanwhile, at Cambridge, two other researchers, an American postdoctoral biologist named James Watson and a British physicist turned biologist named Francis Crick, were trying to solve the same structure, but largely through model building rather than original X-ray data of their own. They built and rebuilt physical models out of metal sheets and wire, cutting cardboard shapes for the bases and twisting thin metal rods into place to represent the sugar phosphate backbone, trying configurations that fit what was known about the molecule's chemistry, and then knocking the whole thing apart again when it failed to satisfy some basic rule of chemical bonding. Crick later described their working method, in his own memoir, What Mad Pursuit, as a process of trial and error conducted almost entirely through physical models rather than careful new measurement, a style of working that some of their colleagues, including Franklin, found undisciplined, even a touch reckless, compared to the painstaking data-first approach of the King's College Lab. There was also a third competitor working in California, the formidable Linus Pauling, already a Nobel laureate, who had recently revealed the structure of a protein component called the alpha helix through exactly this kind of careful model building, and was now turning his considerable talents toward DNA. In early 1953, Pauling proposed a structure, a triple helix, with the phosphate backbone twisted into the center and the bases pointing outward. It was elegant, and it carried the prestige of Pauling's name behind it. It was also wrong and contained a basic chemical error. The phosphate groups, as drawn, would not have been electrically stable and would have required hydrogen atoms that simply weren't there, an error that more careful eyes would soon catch. Watson, upon seeing a draft of Pauling's paper, sent informally to Cambridge by Pauling's own son, reportedly recognized the mistake almost immediately, and the realization injected fresh urgency into the race at Cambridge. If a scientist of Pauling's stature could publish something so close and still be wrong, the prize was clearly still open and clearly running out of time. In January of 1953, Wilkins showed Watson an X-ray image that Franklin had produced the previous year, an image that came to be known as Photo 51, taken under Franklin's direction by her graduate student, Raymond Gosling. Franklin had not given permission for this image to be shown to Watson, and there has been extensive historical discussion in the decades since about how appropriate that was. Photo 51 displayed a striking symmetrical X-shaped pattern, a signature to a trained eye of a helical structure. Around the same period, Watson and Crick also had access, through an internal Medical Research Council report, to unpublished data Franklin had generated, including her calculations of the molecule's helical dimensions. Watson later wrote candidly, in his memoir, The Double Helix, about the excitement and the ethical murkiness of that moment. By his own account, the instant he saw the X-shaped pattern, the helical structure clicked into place in his mind. Franklin, working independently and methodically, was also closing in on the same answer through her own data, but had not yet published. Using these insights alongside Chargaff's earlier chemical findings, Watson and Crick built their final model. The chemist Irwin Chargaff, working in New York in the late 1940s, had spent years carefully, almost obsessively, measuring the exact proportions of the four bases in DNA samples taken from a wide range of different organisms: cattle, yeast, bacteria, human tissue. What he found was a discovery that turned out to be essential, hiding in plain sight inside thousands of careful measurements. In any sample of DNA, regardless of the species it came from, the amount of the base adenine always closely matched the amount of thymine, and the amount of guanine always closely matched the amount of cytosine, a quiet numerical regularity now called Chargaff's rules. The overall ratio of A and T to G and C could vary enormously from one species to another. But within any single sample, those particular pairs balanced out with remarkable consistency, almost as if some invisible bookkeeping rule were enforcing the match. Nobody at the time, including Chargaff himself, fully understood why those particular pairs balanced out so precisely. Chargaff reportedly met with Watson and Crick in Cambridge in 1952, and, by some accounts, found the two young scientists somewhat underinformed about basic chemistry compared to his own painstaking standards. A meeting Chargaff later described with some lingering irritation in his memoirs. And yet it was Watson and Crick who finally connected Chargaff's numbers to a physical explanation. They realized the answer. The bases paired with each other, adenine always reaching across to thymine, guanine always reaching across to cytosine. Like two strands of a twisted rope ladder, where each rung was made of two matching halves, each pairing held together by a small number of hydrogen bonds, gentle enough to be pulled apart when the cell needed to copy itself, sturdy enough to hold the whole structure stable in between. The molecule was a double helix, two intertwined chains running in opposite directions held together by these specific base pairings, coiling around a shared axis like a spiral staircase built for two. On the 25th of April, 1953, Watson and Crick published their findings in the journal Nature, in a paper remarkable for its restraint, barely more than a page, ending with a single understated sentence that has since become one of the most quietly famous lines in the history of science, noting that the specific pairing they had postulated immediately suggested a possible copying mechanism for the genetic material. In the same issue of Nature, Wilkins and, separately, Franklin and Gosling published their own X-ray data, providing crucial supporting evidence for the structure Watson and Crick had proposed. But here's the thing about this moment in the story that deserves to be said plainly without rushing past it. Rosalind Franklin did not live to see how the story would be remembered. She left King's College not long after, by most accounts, relieved to be away from a working environment she had found difficult and at times openly hostile, and continued important work on the structure of viruses, including the tobacco mosaic virus and poliovirus, at Birkbeck College in London under the physicist J.D. Bernal, a scientist with whom she found a far more collegial working relationship. Colleagues from that period describe her as exacting, direct, and deeply serious about the precision of her data, but also warm with the small team of students she supervised, several of whom remained devoted to her for the rest of their lives. She died in 1958 at the age of just 37 from ovarian cancer, a disease some historians have speculated, though without firm proof, may have been connected to her prolonged exposure to X-ray radiation during her crystallography work. She continued, working, by most accounts, almost up until the end. In 1962, the Nobel Prize in Physiology or Medicine was awarded to Watson, Crick, and Wilkins for the discovery of the structure of DNA. The Nobel Prize is, by its own rules, never awarded posthumously, and so Franklin, whose data had been essential and who was never even nominated while she was alive, was left outside a recognition she had quietly, rigorously helped make possible. It is worth noting, too, that history does not record her as bitter about King's College or about Watson and Crick in the way later retellings sometimes imply. By the accounts of friends and colleagues, she had by the time of her death formed a cordial, even friendly, relationship with Crick and his wife. The myth of a simple villain and a simple victim tends to flatten what was, in reality, a more complicated set of working relationships, ambitions, and circumstances. In the decades since her death, historians of science have worked to restore a fuller, more accurate account of her contribution, and her name now sits deservedly alongside theirs in how the discovery is taught and remembered. It's worth letting that settle for a moment. Here in the dark, discovery is rarely as clean as the story we tell about it afterward. It is usually closer to several people, in several rooms, working with incomplete information, racing each other, sometimes generously sharing, and sometimes not, until one combination of insight and evidence locks into place. The double helix was not found by two men alone. It was assembled from Chargaff's careful counting, Franklin's painstaking photographs, and Watson and Crick's willingness to keep rearranging cardboard and wire until the pieces clicked. And once the structure was known, an obvious and beautiful question opened up immediately. If DNA really was the molecule of heredity, how did a cell copy it accurately enough to pass it down, generation after generation, without the information slowly falling apart? Watson and Crick's own paper had hinted at an answer, that single understated sentence about a possible copying mechanism. The double helix's elegant base pairing suggested that if you simply unzipped the two strands down the middle, each old strand could serve as a template for building a brand new partner strand, with adenine drawing thymine into place, guanine drawing cytosine. This would mean every new DNA molecule would be a mix of one old strand and one freshly built strand, a model called semi-conservative replication. But it was only a hypothesis, an elegant guess. Other models were also still possible. Perhaps the whole original molecule stayed intact and a wholly new copy was built alongside it. Or perhaps the strands got chopped up and reassembled in some more scrambled way. The experiment that settled the question, conducted in 1958 at the California Institute of Technology by Matthew Meselson and Franklin Stahl, is still described by many biologists as one of the most beautiful experiments ever performed. Elegant, not because it was complicated, but because of how cleanly its result mapped onto a clear, simple prediction. Meselson and Stahl grew bacteria in a medium containing a heavy isotope of nitrogen, so that the bacteria's DNA became measurably heavier than normal DNA. Then they switched the bacteria over to a medium with ordinary, lighter nitrogen and let them divide. Generation after generation, carefully extracting and weighing the DNA at each step using a technique called density gradient centrifugation, which could separate molecules by the tiniest differences in mass, spinning samples at extraordinarily high speeds until even fractions of a percent of weight difference settled into visibly distinct layers. If replication were semi-conservative, the first generation of new DNA should come out at an exact intermediate weight, half heavy, half light. And that is precisely what they found. By the second generation, the DNA split cleanly into two bands, one intermediate and one fully light. Again, matching the prediction perfectly. Watson and Crick's elegant guess turned out to be exactly correct. The cell really does pull the double helix apart like a zipper and use each old strand as a template for a new partner. But this answer, satisfying as it was, opened up yet another question. Almost immediately, the way a single unlocked door in this story always seemed to reveal another hallway behind it. If copying really happened this way, strand by strand, letter by letter, billions of times across a single lifetime, and trillions of times across a single lineage, then surely, every so often, the copying machinery would make a mistake. A wrong letter slipped into place, a tiny stretch skipped or duplicated. And it does. These copying errors, called mutations, turned out to be one of the central engines of evolution itself, the raw material upon which natural selection acts, the reason no two siblings are genetically identical, the reason a virus can shift its disguise from one flu season to the next, the reason in cancer can arise when the wrong mistake lands in the wrong gene at the wrong moment. Suddenly, the molecule that explained how traits were passed down faithfully from parent to child also explained how those traits could occasionally, quietly, change. And in that quiet unreliability, built into a system designed for precision, sat the entire mechanism by which life on this planet had diversified across 3.5 billion years, from the first simple cells to every form alive today. So now scientists understood the shape of the molecule, and they understood how it copied itself. But there was still a deeper mystery sitting underneath all of it. If DNA was a code, a sequence of 4 chemical letters, A, T, G, and C, what exactly was it a code for? How did a string of 4 repeating letters translate into the astonishing complexity of a living, breathing organism with its proteins, its enzymes, its structure, its function? Cracking that code became the next great race. And by the late 1950s, it had attracted some of the sharpest minds in biology, including, in an unusual detour, a small informal group that called itself the RNA Tie Club, which counted the physicist George Gamow and, for a time, even Watson and Crick themselves among its members. Scientists trading ideas about how a 4-letter alphabet might be translated into a 20-letter one, decades before anyone had the experimental tools to test their guesses directly. It reached its first real breakthrough in 1961 in a modest laboratory at the National Institutes of Health near Washington, D.C. Marshall Nierenberg, working with a German postdoctoral researcher named Heinrich Mattei, designed an experiment using a synthetic strand of RNA made of nothing but repeating units of a single base, uracil, written as a long string of U, 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 U. When they added this synthetic RNA to a cell-free mixture capable of building proteins, the resulting protein chain was built entirely out of a single amino acid, phenylalanine. The implication landed with startling clarity: a repeating sequence of 3 U's, a codon, as it would come to be called, corresponded to the instruction phenylalanine. For the first time, a piece of the genetic code had been definitively read, not guessed at or reasoned toward, but directly demonstrated in a test tube. Nierenberg, by most accounts, a quiet and somewhat unassuming scientist working outside the more fashionable centers of molecular biology at the time, first announced the finding at an international conference in Moscow in 1961 to a room that reportedly fell into a kind of charged, attentive silence as the implications became clear. From there, the race accelerated. Other scientists, notably Har Gobind Khorana, working with synthetic RNA sequences of greater complexity, and Robert Hawley, who worked out the detailed structure of transfer RNA, the molecule that ferries amino acids to the protein-building machinery, helped fill in the rest of the picture. Within a few years, the entire genetic code had been worked out. 64 possible three-letter codons, translating into the 20 amino acids that build every protein in every living thing on Earth, plus a handful of codons that signal where a protein building instruction should start and stop. Nierenberg, Khorana, and Hawley shared the Nobel Prize in Physiology or Medicine in 1968 for this work. The code, it turned out, was almost universal. Astonishingly, nearly the same three-letter dictionary is used by bacteria, oak trees, jellyfish, and human beings alike. A quiet signature of how deeply connected all life on this planet really is, tracing back to some shared ancestor billions of years ago. And here's where it gets interesting in a slightly different way. Once scientists could read the code in principle, they still needed a practical way to read it in any particular stretch of real DNA, to take an actual gene from an actual organism and determine its exact sequence of letters, one by one. That problem belonged largely to a quiet, meticulous British biochemist named Frederick Sanger, a man who had already won one Nobel Prize in 1958 for working out the complete amino acid sequence of the protein insulin, itself a landmark achievement that took him over a decade of slow, careful chemical detective work, fragment by fragment, with no shortcuts available at the time. Sanger was, by the recollection of nearly everyone who worked alongside him, an unusually modest man, more unusually modest man, more interested in solving the puzzle in front of him than in claiming credit for it, and famously uncomfortable with public speaking and self-promotion even after he had become one of the most decorated scientists alive. In the mid-1970s, working at the Laboratory of Molecular Biology in Cambridge, Sanger developed a technique that became known as the chain termination method, or simply Sanger sequencing. It used specially modified building blocks called dideoxynucleotides that, when incorporated into a growing DNA strand, would stop the strand from growing any further, like a typo that halts a sentence mid-word, a missing connector where the chain expected to keep building. By running 4 parallel reactions, each one set up to terminate at a different one of the 4 bases, and then sorting the resulting fragments by length on a thin gel, using an electric current to pull shorter fragments further than longer ones. Researchers could read the underlying sequence directly off the resulting pattern, letter by letter, almost like reading rings in a tree trunk. In 1977, Sanger's team used this method to sequence the entire genome of a small virus called bacteriophage phi X174, 5,375 bases in total, the first complete genome of any organism ever sequenced. Sanger received a second Nobel Prize in Chemistry in 1980 for this achievement, shared with Walter Gilbert, who had developed a related sequencing method, and Paul Berg, recognized for his pioneering work in recombinant DNA technology. Sanger remains, to this day, one of only a handful of people in history to have won 2 Nobel Prizes, and reportedly remained quietly bemused by the attention this brought him for the rest of his life, eventually retiring from active research altogether to spend his time gardening. Sequencing technology kept improving through the 1980s, but reading an entire human genome, some 3 billion base pairs, spread across 23 pairs of chromosomes, still seemed for a long time like an almost unreasonable ambition. To put that scale into some kind of perspective, if every base pair in a single human genome were printed as a letter on an ordinary page, The resulting document would run to roughly 1 million pages, a stack of paper taller than most office buildings. And yet, in 1990, an international collaboration formally launched exactly that project. The Human Genome Project, coordinated primarily through the United States National Institutes of Health, with Francis Collins eventually directing the effort, alongside the Department of Energy and research centers across the world, including the United Kingdom, France, Germany, Japan, and China, set out to sequence and map every gene in human DNA. It was, at the time, one of the most ambitious scientific undertakings ever attempted. With an original projected timeline stretching across roughly 15 years and an estimated cost across its full life in the range of several $billions. A scale of public scientific investment that invited its own share of skepticism in the early years, before the practical and medical payoff had become clear to skeptics in government and the broader public alike. The project gained an unexpected jolt of competitive energy in 1998 when a biologist named Craig Venter, who had earlier worked at the National Institutes of Health himself, founded a private company, Celera Genomics, and announced his own, faster approach to sequencing the human genome using a technique called whole genome shotgun sequencing, breaking the genome into countless small fragments, sequencing each one with newly automated machines descended from Sanger's original method, and using powerful computing to reassemble the fragments by matching overlapping ends, rather than methodically working chromosome by chromosome as the public project had originally planned. The ensuing competition between the public consortium and Venter's private effort became, for a few tense years, something close to a public spectacle, covered in newspapers and magazines the way a sporting rivalry might be. And it pushed both sides to accelerate dramatically, refining their machines and their methods at a pace neither side might have managed working alone. In June of 2000, President Bill Clinton and Prime Minister Tony Blair jointly announced, via a live satellite link between Washington and London, that a working draft of the human genome had been completed. A moment Clinton described as akin to learning the language in which God created life, language that, however poetic, reflected the genuine sense of historical magnitude. Many scientists felt in that room. The full, refined sequence was declared essentially complete in April of 2003. Fittingly, exactly 50 years after Watson and Crick's original Nature paper. What the Human Genome Project revealed was, in some ways, humbling. Scientists had long assumed humans, with all their biological complexity, must possess an enormous number of genes. Some early estimates ran past 100,000, based on a rough assumption that more complexity must require more individual instructions. The actual count turned out to be far smaller, somewhere in the range of 20,000 to 25,000 protein-coding genes, not dramatically more than a fruit fly, which carries something in the neighborhood of 14,000, and fewer than some species of rice, a plant that, by simple gene count alone, is genetically more elaborate than the human body that might eat it for dinner. It was a finding that genuinely surprised much of the scientific community when it was first confirmed, and it forced a quiet but significant rethinking of what complexity in biology actually meant. The complexity of a human being, it turned out, wasn't simply a matter of having more genetic ingredients, but of how intricately those genes were regulated, switched on and off, combined and recombined, spliced into different versions of themselves depending on context, across different cells and different moments in development. A layer of complexity scientists are still working to fully understand even now, decades after the genome itself was declared complete. The story doesn't stop at reading the code. Once scientists could read DNA, the next ambition, almost inevitable, in hindsight, was learning to edit it. An early and crucial tool arrived in 1983, when a biochemist named Kary Mullis, working at the Cetus Corporation in California, conceived of a method called the polymerase chain reaction, or PCR, which allowed scientists to take a tiny, almost vanishingly small sample of DNA and copy it millions of times over in a matter of hours, using cycles of heating and cooling, and a heat-resistant enzyme borrowed, fittingly, from bacteria that live in the scalding waters of hot springs, organisms that had themselves been collected and studied for entirely unrelated reasons years earlier. The method essentially automated and dramatically sped up the same basic zipper and rebuild process that Meselson and Stahl had confirmed decades before. Except now a scientist could trigger it, again and again, inside a small machine on a benchtop, doubling the amount of target DNA with every cycle, so that a sample too small to detect at the start of an afternoon could become abundant enough to study in detail by evening. Mullis has described the core idea arriving to him, by his own account, during a nighttime drive along a California highway, the structure of the solution apparently assembling itself in his mind somewhere between mile markers. A story that has become part of scientific folklore, even as the surrounding details have been retold in various, sometimes embellished, ways over the years, including by Mullis himself, who remained, throughout his life, an unusually colorful and unconventional figure, even by the standards of scientists known for eccentricity. PCR transformed genetics, forensics, and medicine almost overnight, turning what had once required painstaking, large-scale samples into something that could be done with a single hair follicle or a drop of blood. Mullis received the Nobel Prize in Chemistry in 1993 for the discovery. The most recent chapter in this long unfolding arrived through, of all things, an obscure feature of bacterial immune systems, first noticed almost by accident. Scientists studying the genomes of various bacteria back in the late 1980s and through the 1990s had repeatedly noticed strange repeating patterns turning up in the sequences they were reading. Short, near identical stretches of DNA, evenly spaced apart by unique segments in between, like beads on a string, with the spacing always coming out suspiciously regular. For years, nobody quite knew what to make of the pattern, and it sat in the scientific literature as a documented curiosity rather than a solved mystery. It eventually came to be called CRISPR, an acronym for clustered, regularly interspaced, short palindromic repeats. The breakthrough in understanding what these repeats were actually for came when researchers realized that the unique segments tucked between the repeats matched almost perfectly the genetic sequences of viruses known to infect bacteria. It turned out bacteria use these sequences, together with associated proteins, to store snippets of viral DNA from past infections and use them to recognize and cut up that same virus if it ever returns, a kind of molecular memory of past threats. Assembled one infection at a time, across countless bacterial generations, a microscopic immune system with its own working archive, not unlike In its own small and ancient way, the very molecule this entire story has been circling around from the start. In 2012, the biochemist Jennifer Doudna at the University of California, Berkeley, and Emmanuel Charpentier, then working in Sweden and France, published groundbreaking work showing that one particular bacterial protein in this system, called Cas9, could be reprogrammed with a custom-designed guide sequence to cut any targeted stretch of DNA scientists chose, turning what had been a bacterial immune defense into a programmable, precise gene editing tool, far cheaper and more accessible than anything that had existed before. Other researchers, including Feng Zhang at the Broad Institute, made important subsequent contributions to adapting the system for use in more complex cells, and the question of credit and patent rights surrounding CRISPR became, in the years that followed, the subject of extensive and at times contentious scientific and legal debate. In 2020, Doudna and Charpentier were jointly awarded the Nobel Prize in Chemistry, the first time a Nobel Science Prize had been awarded to a team made up entirely of women. That power, of course, arrived with new and difficult questions attached. In November of 2018, a Chinese researcher named He Jiankui announced that he had used CRISPR to edit the genomes of human embryos, resulting in the birth of twins, in an attempt to make them resistant to HIV, a claim that triggered widespread alarm throughout the international scientific community. The experiment was conducted with what most experts agreed was insufficient oversight, unclear consent, and unknown long-term risk. And he was widely condemned by scientists across the world, eventually facing legal consequences in China. The episode became a sobering reminder that the same tool capable of correcting disease at its root could also, if used carelessly, alter the heritable genetic code of a person before they were ever born, in ways no one could fully predict or take back. It pushed much of the scientific community toward firmer, shared guidelines about where the line should sit between treating disease in an existing person and editing the heritable genome of someone not yet able to consent to that choice at all. There was also, by this point in the story, a quieter and equally humbling realization happening inside the genome itself. For years after the Human Genome Project's completion, a great deal of the human genome, by some estimates, the vast majority of it, appeared, on first inspection, to do nothing at all. It didn't code for any protein. Some researchers, only half-joking, took to calling it junk DNA. But as sequencing technology improved further through the 2000s and 2010s, and as a large follow-up international effort called the ENCODE Project began systematically cataloging what all that supposedly empty space was actually doing, that label began to look like another version of the same old mistake Levin had made a century earlier, assuming that because something looked simple or purposeless, it must be unimportant. Much of this so-called junk turned out to be deeply involved in regulation. Switches, dimmer controls, and timing mechanisms that determine when and where particular genes get turned on or off, why a liver cell and a skin cell can carry the exact same genome and yet look and behave nothing alike, why the same set of instructions can build a far more elaborate result in one context than another. An entire field called epigenetics grew up around the discovery that chemical markers sitting on top of the DNA sequence, small molecular tags added to the DNA itself or to the proteins it winds around, could influence which genes get read without changing the underlying four-letter sequence at all, somewhat like sticky notes and underlining added to a book rather than rewriting its actual words. Researchers studying identical twins who begin life with exactly matching DNA sequences found that as those twins aged, their epigenetic markers gradually diverged, shaped by years of differing diet, stress, environment, and experience. A slow drifting apart that helps explain why two people who started out genetically identical can, decades later, develop quite different patterns of health. In some carefully studied cases, certain epigenetic markers have even been shown to persist across a single generation, passed from parent to offspring in modified form, though the extent and permanence of that kind of inheritance remains an active and still debated area of research. The deeper scientists looked, the more it became clear that the genome wasn't simply a flat list of ingredients. It was something closer to an enormous, layered script, with stage directions as important as the dialogue itself. Think about the distance traveled across that long chain of discovery. From a young man scraping nuclein out of pus-soaked bandages, unsure what he'd even found, to tools that can now, with some real precision, locate a single misspelled letter among 3 billion and correct it. From a quiet monk counting peas in a monastery garden, with no audience and almost no recognition in his own lifetime, to a technology capable of editing the living code of a cell. Each discovery sat quietly until someone else picked it up and carried it one step further, often without knowing exactly where the next step would lead or who would take it. And this brings the story, gently, back toward tonight, toward where you are now, lying still, your own DNA folded inside the nucleus of essentially every cell in your body, copying itself patiently as old cells are replaced by new ones, all without requiring a single thought from you. The science born from those bandages and those pea plants now touches ordinary life in ways that would have seemed like fantasy to Miescher or Mendel. A simple swab of saliva mailed off in a small plastic tube can now trace ancestry back across continents and centuries, connecting distant cousins who never knew of each other's existence, built entirely on the same base pairing principle. Chargaff first noticed in his careful counting. A blood test can screen for inherited conditions before a child is even born, giving families information that, only a few decades ago, no test on Earth could have offered them. Gene therapies, still young, but advancing quickly, are beginning to treat certain inherited diseases, conditions like sickle cell disease and certain inherited forms of blindness, by correcting or replacing the faulty instructions at their source, rather than only managing the symptoms those instructions produce. One such therapy, approved for clinical use in the early 2020s and built directly on the CRISPR system first described by Doudna and Charpentier, works by editing a patient's own blood-forming cells outside the body before returning them, switching on a fetal form of hemoglobin that the body normally silences after infancy, and offering some patients with sickle cell disease relief from a condition that had caused them lifelong pain since childhood. It is a strange and moving thing to consider that the same base pairing logic Chargaff first noticed in his careful counting, and the same bacterial defense system once studied only out of pure curiosity, now sits inside an approved medical treatment, quietly easing suffering in hospitals around the world. Forensic science, transformed first by PCR and then by increasingly sensitive sequencing, can identify a person or clear an innocent one from a fragment of evidence too small to see with the naked eye. A single hair root, a trace of saliva on the rim of a glass. None of this existed, even in rough outline, within the lifetime of your own grandparents, and some of it has only existed for less than a decade. And yet, underneath all of that astonishing modern capability, the fundamental story stays almost unbearably simple, almost quiet. Uh, When you sit with it the way you're sitting with it now, 4 letters, a twisting ladder, a zipper that pulls apart and rebuilds itself, strand by strand, inside nearly every living cell on Earth, including, right now, the ones in your own resting body. The same 4 letters that built jellyfish and oak trees, and the dinosaurs whose bones you might have seen in a museum once as a child, are the 4 letters quietly, endlessly copying themselves inside you tonight, with no urgency, no effort, no need for you to be awake for any of it. So let that be the last thought you carry, loosely, without needing to hold on to it. Somewhere inside you, in the dark and the quiet, something ancient is simply continuing, the way it has continued, unbroken, since long before there were names for any of it, since long before there was anyone awake to wonder about it at all. You don't have to follow that thought any further than this. You don't have to follow it anywhere. Just let your breath find its own slow rhythm now, the way it always does without your help. Let your body grow heavier into whatever is holding it. The blankets settling, the room quiet. Nothing left to figure out, only this, soft, still, here,